few words to welcome, but Jay, you can also.
I think Jay is not on board.
Jay just,
Yet to
well,
join, I think.
maybe Jay has left the meeting. Well, so as everybody can see, I've started recording this meeting because not everyone has attended yet. Welcome everybody, welcome all ambassadors to this bioinformatics meta-analysis of the ESKAPE pathogens. From what I remember from the previous meeting is that we have all made a lot of progress, a lot of work has been done, a lot of inspiring work. By the way, when I was in the US, I also mentioned to some people the idea that we are doing this, and I got some very enthusiastic responses, I can tell you. Um, Last time, we also decided to make some additional standard operating procedures, video for instruction. These have been shared with all of you, and I think Krishna, it's up to you now to well give a follow up on this and what you want to share during this meeting.
Yeah. Thank you, Martin. Thank you, Martin, for welcoming everyone, and also welcome all our ambassadors on board to this meeting. And we had a meeting, I think, around 15 days ago with respect to the what's the next step that we are going to take up with these data. We really thank all the ambassadors for running the abrogate to get various kinds of resistant determinants, virulence determinants, and the plasmids. That's from a huge number of data that we have started with. So really appreciated the hard work that you did in spite of your busy schedules with your routine work. So that as a part of the next step, how we want to take it further. So we are during the analysis process, we collected the metadata as well as we have run the abrogate to get resistant determinants. So the next task is to harmonize these data and make the metadata perfect and map the metadata with the existing the determinants, reverse determinants that we have. So that's the next step. So to do that, myself and the Prithvi, we have done an effort to prepare an SOP, and these SOP details how exactly the step of the metadata cleaning as well as resistant determinant needs to be analyzed further. So that is what we have done it in the previous meeting. And as a result of that, we made a video SOP as well as the PDF document that has already shared. So then in this particular meeting, we would like to hear from you. Do you have any particular doubt with respect to the cleaning the data right now that we have? Because when we see the metadata has, it's full of many things are there, but that is not actually necessary. And some of the data that needs to be updated as which is necessary for our analysis. For example, first thing is about the locations. So the locations, they have mentioned the country locations, but if we see the country, maybe we will be have the skew data. So that's why we have planning to go for a continent wise. So now our job during the metadata cleaning is to map those country to the respective continents so that that data will be uniformly added. The second is with respect to the date when this particular isolate was collected. So right now we have it is in DDMMYY format, that is date, month, and year. So all those date, month, and year that will be normalized to only to the year. So we don't interested in the date and month. So we will make it uniform. So that's the one thing. So how to do that? So for that we have done an SOP and that we have shared with you all. So and once the metadata has been cleaned, the next task is another component is these isolates what we have analyzed, which has come from different sources. It could be from the clinical sources, or it could be from the environmental sources, or it could be from the animal sources, or from the plant sources. So the appropriate classifications we have to make. So what are the appropriate classification we can make? There are 4 classifications that I already shared with the SOP. So according to the samples that from which this genome has been retrieved, accordingly you can put it under those categories, and you can denote that. So these are the 3 or 4 important tasks that we need to carry out during this metadata cleaning. Once the metadata has cleaned, the next task is to merge the metadata with the resistant determinant that is obtained from the abrogate. So that now we will be having the full-fledged data in our hand to do the next, the statistical analysis. The statistical analysis, I believe that the video that has been previously shared has those details. What are the statistical analysis we are going to conduct? Once the metadata is ready, again, we will have one more meeting to actually see how exactly we will represent our data. First, continent representation. The second, year-wise trend. Is there any differences in the year-wise trend in the resistance pattern? Then the third, there are 4 segments, as I mentioned: clinical segments, animal segment, plant segment, and environmental segments. So in these 4 segments, how the resistance patterns are behaving. So that's the 3 or 4 different perspectives that we are going to add on to our data to make more meaningful results from those analyses that we are going to carry out. So this is what we have tried to put it in these the PDF and the video that we have shared with you. And I really appreciate the Prithisagar's effort in doing this SOP and the video demonstration. So in this meeting, if you have any doubts, we can clarify. And all ambassador, we will put together our effort to make this particular more interesting with your inputs, along with the expert opinion from Martin and Jay. Over to our ambassadors to clarify and add any suggestions, input. Hi, Jay.
Hello,
Yes.
Jay.
Hello, Martin. Hello. Yeah, everyone. I'm sorry for the late because the system went off and it restarted again. I don't know why. And
yeah,
I tried
during
to
your
join.
absence, we have discussed about what actually the metadata cleaning SOP has, and
Okay.
what we are expecting from those metadata cleaning. So that is what
Yes.
I have detailed
Okay.
here. And the SOP has been shared, and the video demonstration has been also given to all the ambassadors.
Yes.
So during this meeting, we will clarify any doubts they have, or
Yes.
if they want to provide any inputs to us, we can also try to accommodate those inputs in our SOP to make the study more meaningful and more scientific.
Yeah, perfectly okay. I think that's the one we really wanted, the clarification and other things. Yeah. Please proceed. I see. 6 5 5 6. Yes.
I think most of our ambassadors are there, except few. Yeah.
Yes.
We have a
Bruno.
good number. Good
Yeah,
representation.
Saranya may not be joining because she's in home. She's traveling for some urgent reason.
Okay. Yeah.
So, but she will be updated. Okay, good. Okay.
Yeah, we are making a recording, Jay.
Yes. Recording
So
should be.
She will get a recording of this meeting.
Wonderful. That's really nice of you.
So now the forum is open for discussions, the clarifications, and inputs,
And
those
suggestions.
things, and
That's
suggestions.
very important.
Yes.
Okay.
I hope that everyone has gone through the SOP as well as the video demonstration that we have shared.
No, sorry, not yet, because last week was very busy and I just received the email from Martin regarding all the SOPs and cleaning mechanism, but I could not perform. I have all the material downloaded, but I have not yet opened this video because it was a busy week, and I was not expecting this meeting. With some results in hand. So, but sure that there will be no problem, and we will clean the data very soon.
Okay.
No issue. I think this particular meeting we have conducted just to make sure that everyone has received the SOP and video demonstration. If there is any clarification with respect to handling and doing that, so we will try to answer that, as well as we will take your suggestions and inputs.
Yeah, the suggestions to improve is also
Suggestions to improve. Yes.
Yeah, because this kind of work we can really do a lot, and then we can abridge this work. I think Martin, if you are, everyone agree, we can give one try to Lancet microbe.
Can you say it again, Jay? What would you say?
We can give one big try to Lancet microbe.
To Lancet microbe, but
Lancet.
Jay is telling once the results is ready, we can try to publish in Lancet journals.
Oh yeah, in the Lancet. Okay, yeah, yeah.
Yes.
Sorry, the journal, the Lancet. Oh, that would be great.
Not the Lancet, original Lancet. We can go for Lancet Microbe.
Microbes.
Oh, Lancet microbes. Okay, that is
Yeah,
okay. Yeah,
that's
sure.
specialized.
Great.
That's specialized. What we
Lancet.
can do is that with the AMR group and with this ESKAPE pathogen, which is very important, I think, Martin, you can design one wonderful cover letter and we can mesmerize them.
Yeah. Oh, happy to do so. I already, you were not. I told in the beginning already, Jay, when you were not joining us, that I've told about to several people in the United States that we were doing this project, and I got some very enthusiastic reactions. So this is great work. Yeah.
Okay, yeah. And your proposal for an internal reviewer is also very good.
Yeah.
Yeah, we met this colleague in the forum long time
Yeah.
back. Not so long time back. He was a
Yeah.
nice guy, and
He's a
he
nice guy.
knows
Yeah.
know-how because he's a mathematician and
Yeah.
turned to be a thing. I think that's a good proportion. I immediately discussed with Krishna, and Krishna is also happy to
Yeah. Yeah.
accept this.
Yeah.
Yeah.
Okay. Yeah.
I think we can have an internal discussion and we can really fight for this. Of course,
Yeah.
there are miles to go, but still we can have an aim. If this is successful, we will reach a
Absolutely.
lot. We'll reach a lot.
Absolutely. Absolutely. Yeah. Yeah.
And you know the impact factor of Lancet microbe, it's something like 86
It's
or
80.
something.
It's 86. Yes, because
86.
I mean, I submitted a paper in Lancet Infectious Diseases 2 weeks ago, and today they offer me to transfer this to the Lancet Microbes. So
Oh,
I said okay, fine.
we can really tell yes.
I mean, they
Great.
actually they rejected from Lancet Infectious Diseases. It was reviewed by five reviewers, and then two were agreed and two were not agreed. So eventually, I mean, the editor decided to offer me the transfer to Lancet Microbes. So now it is in Lancet Microbes. Let's
Yeah.
see. Let's see.
Hopefully,
luck.
hopefully it will pass through. Best wishes,
Yeah.
huh?
Good luck. Good luck.
And the good idea, and thank you, and the good idea maybe is to go for a clinical microbiology review. I mean, because we have a review article, so it's like a meta-analysis and review. We can also opt for clinical microbiology review, which is like 50 impact factor nowadays.
No,
It's
this one is not a review. This is a research article. That's why I don't
yeah,
want to go.
but they do publish meta-analysis.
Okay, okay,
Yeah,
yeah. But Lancet microbe, we can give one big try when
yeah.
everyone
Why not?
agrees.
Yeah.
Why not?
I agree.
Because everyone agrees, then we can give a big try. We can incorporate. Of course, this internal reviewer also. We can also ask him to come into the picture because this internal reviewer also has a Lancet microbe. Today morning I read it. He also has a paper in Lancet microbe. So he knows know-how, know-how to convince the things, and Martin can write a nice
Yes.
polished
Sure. Yeah.
I
Super.
mean, cover letter.
I will
Okay,
write a
so
super
we'll
nice
come
letter.
for this later when we got all these results.
Yeah.
Are there any questions from any of the partners or any of the things, or any suggestions? Or if anything is not clear, of course, you can also always reach us in the WhatsApp group.
Yeah, yeah. I have only one concern because we submitted a meta-analysis last week in clinical microbiology review, but they rejected, and then we switched to microbiology spectrum. So the thing they were asking that what was the meta-analysis was reviewed by some independent, as you said, internal reviewer by independent reviewers, and the data should be validated. For example, the data what we have should be validated by a blind reviewer from within the group, or maybe two authors from within the group.
Okay.
And they also demanded us statistical lot of statistical inputs. I mean, we did random effect model, RD models for all the meta analysis. We did this nice meta. There is a R package called Metafloor, I think. It's a R package. It is specific for meta analysis. I mean, we did all the analysis on our this metaflow. So maybe if you allow me, I can quickly share the screen.
I think we can, Martin, you can allow him so that.
Yeah, sure. You can, Moscow.
Okay. So maybe quickly I can show you that. It's fine. So are you able to see?
Yes.
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 Yeah,
Yeah,
okay.
we are able to see that.
Great.
Yeah.
Okay, maybe I think this one is figures. Yeah.
You're trying to open a PDF document that also we see.
Yeah,
Okay.
exactly. So this is what we did in our meta-analysis. I mean, it is currently under review. So we search many articles from Web of Science and PubMed, and then we screen all these articles, and finally, so this is this is the picture what they want to see from meta-analysis.
Okay.
Yeah, and for that, I mean, one of my student did all these analysis on R package, and there we see, for example, this prevalence in terms of the weight of the study. The larger the sample size, the more the weight, and then the confidence interval, and then we have some specific subgrouping. For example, this is E. coli subgroup. It has its own random effect model at the end, and then we have Klebsiella pneumonia group, where you have again random effect model for this subgroup. I mean, this is all done on R package. So I mean, maybe we need this. We have a lot of, and then we can see the incidence rate on this at the bottom. And overall you can see that you can see it's like 9% prevalence rate of whatever we are looking for. Similarly, so we have another subgroup, and then another subgroup. Maybe we can divide over.
I see 17 pages, sir.
Yeah. Many pages, many. I mean,
17 pages totally.
Yes, we have a lot of picture from Pakistan, and then the data is showing where we are. And then clinical, we have subgroup like clinical, and then like animal or food isolates. So we have a lot of subgroupings here. Some are like NDM positive. Then there is another group, and then I can show you also this Circos diagram where we try to map all the genes, and then with the isolates, what we have on the right side we have isolates, and on the left side we have these resistance genes. So similarly, we also compared with the sequence types. So we have on the right sequence type, and on the left of this figure we have the genes like NDM1 gene or whatever. So we did a lot of we are only 2 people in this review.
Oh, that's cool. Huh?
Yeah.
Okay.
And
how long he was working on that?
It took us 6 months.
6 months
for the yeah. But if we learn a lot of things. I mean, mainly it's my student. When you have the data in Excel, I mean, then this data analysis is no problem in the R package, because we were learning. This was our first experience. But now, if you can give us the data in the Excel sheet, like in the metadata, we can generate all these type of particularly these random effect models and all these. But first we should know what we are trying to do.
What we are trying to do, exactly. What we are trying to extract from that.
Yes.
So the with respect to our data, right now the data has been cleaned, and all these are in Excel format for Enterobacter. So that's beautifully done by Prithvi. So that's available right now.
Oh, great.
Yeah.
Great.
I think we can use.
Would you like to see that data? If you allow me, I can share that.
I would recommend to so. I would recommend to so. Yes, please.
I'm doing right now. Give me a minute.
Yeah.
Is it visible?
Yeah.
Yeah.
Yes, it's
So
coming.
now
Yeah.
this has been the metadata has been cleaned, and the collection data location and the isolate type has been described properly for these Enterobacter samples. This Enterobacter, I feel it is having around 5,000 genome sequences, and for all the 5,000 genome sequences, this metadata has been cleaned. So if you see the collection date, so collection date has been it's from, I think, give me a minute. Yeah, I'll put the sorting option, and you can see here the collection date is start from 1982 to 2022, and with some unknowns, because some of the isolates we don't have the collection date from when it was collected. So that's why we have put it under the category unknown, and the same information that has been given in the SOP as well. And the second column is the continent, the location from which continent this particular genome has been obtained. So you see here informations are there: Africa, Asia, Europe, Northern America, Oceania, Southern America, and unknown. So these we have also given a UN list for the continent classification that has already shared by Martin with all the ambassadors. So the next column is about the isolation type. If you see the isolation type, as I mentioned, we have 4 different compartments, like animal, that's obtained from genome obtained from clinical sources, environmental sources, and the plant sources. And for some of the samples, we don't have a isolation source. So in that case, we have marked it as source unknown. So on the E column, we have the total number of return minutes that has found in this particular isolate. And in the F column, that we have not detailed with all the ambassadors, we are trying to develop a mathematical calculation by which we are trying to calculate the MDR index, how we have a MAR index for antibiotics, the phenotypic resistance. So we have developing a kind of an algorithm. So this is called as the MDR score or MDR index. This is based on the presence of genome, the presence of genetic elements. So that I'm trying to do that. I think hopefully that will be ready within a few days. So all the next columns are for different antibiotic determinants. You can see here. So it is for the aminoglycoside. I have classified genes into a specific classes. Aminoglycoside C, I have made it here. And you can also have here again aminoglycoside. Now it is cephalosporins. It's 3rd generation cephalosporins, especially the CTX cephalosporins here. Now you also have for the fosmycin, carbapenemase resistance, the colistin resistance. What are clinically important? Only those resistance that I have highlighted in this particular Excel sheet. Lot of effort has went into do the data cleaning. I hope that the same will applicable for all the ambassadors to do the data cleaning of their data that they have. I think they have do that very carefully, and they have to use all those criteria that we have given it in the SOP. So it's just a sample that we have done it for Enterobacter. Yeah, thank you.
Great. Thank you.
Thank you.
I'm afraid for Klebsiella pneumonia we have 36,000 genome.
Exactly. I think yours is the highest.
Yeah.
I think we can after cleaning, when we are doing the process, we can also talk. Give to Muskur student, and we can also try to do the things.
For the next statistical analysis, definitely we will take help from Muskur to plan what are those different statistical analysis we will conduct, because he is expert in R. So definitely.
Yes. We can. He's currently in Oxford. I mean, he's doing PhD, but I mean, he just moved 6 months ago, so we can always ask him, and he will be available for the analysis.
He's in Oxford. I think Prithvi is also in UK now.
He's in Plymouth. Prithvi, are you there today?
Yes, sir.
I saw him.
Yeah, can you switch on your video?
It's on. It's not.
It's on already. Okay.
Yeah.
Okay. Hope you are enjoying the trip, Prithvi.
Yeah.
It's a small world.
Okay. So any other questions or concerns or suggestions? Prithvi, you have some other points to add up with SOP and other things?
Uh, uh, just now, like I was today working with the same data, then sir told me to work on the intrinsic resistance that we have to separate. So I think we have to. I was seeing the UCAST guidelines and the document I just found. So I think that also I think we have to share with the ambassadors so that they can refer that guidelines and separate the intrinsic resistant genes. I just downloaded the file, so I think I'll discuss with her and that also will share. And one more thing is that right now we are working on the card database summary. Since we have run applicate for card, VFDB, and plasmid finder, I think ambassadors should know that they should clean first the card database summary, not VFDB or plasmid finder output, because now, right now we are working on the antimicrobial resistant genes as of now, not the virulence and other things.
Other
So
genes, yeah.
that are the only 2 points just I wanted to make. And anyways, if anyone has any doubts, they can directly contact through WhatsApp. I think we can. I'll try my best to respond them in at the earliest as it is possible. That's all from my side. Thank you.
Yeah, correct. Correct. Okay. Yeah, Dorcas has got some issue, it seems. I will appreciate it. Oh, Staphylococcus group is working. Krishna, maybe you can read it, the chat
Someone
box.
in the group. Yes. Yeah. May I know who is your the collaborator for Staphylococcus? Do you know any idea? I need to check my document to see who is.
File.
Yeah.
All right.
Yeah, Muskur wants to go to another meeting. Yes, Muskur
Yes.
can leave,
Yeah.
of
Yeah.
course.
Yeah.
I think we are also done.
Yeah.
Prithvi, do you have that list with you right now? Can you just check it up and?
Prithvi, your audio is muted.
Krishna, you can probably find in the older emails or the mailings who was in this group.
Yeah, I'll
Okay.
just check in my
And
email.
then involve him or her
Yeah.
to help
I think
Dorcas.
this one
Dorcas.
we can also solve it later also because we can also solve it WhatsApp.
Yeah.
We can try to the partner who is working on
What's the genome number, Dorcas, you have?
Yeah, the genomes are up to, I think, 30,000 genomes.
30,000. Okay.
Oh, that's a huge.
And should we schedule another meeting already now?
I think I have found the. Can I share my screen so that
Yeah.
we can see who it is? So this is how it's been assigned.
Staphylococcus. Yeah. This is Abdullah Abarode. I think this particular ambassador.
I think he has. I think he has dropped off from the project right from the beginning.
Right from the
'Cause
beginning. Exactly.
Yeah,
Exactly.
I remember contacting him on how we are to do the work, but he wasn't responding. I
Okay.
contacted him twice, but he wasn't showing me positive response. So I had to do the whole work.
Yeah.
So, but I think for further analysis, I will need assistance actually.
Definitely, we will try to help you. Prithvi, is it possible for us to help Dorcas?
I think we can help also, like if someone else is coming on board for this team, because if some member is not there, then if like the Mashkur student, if he is also coming on board, maybe we can assign him also for
Yes, we will help. We will help the Dorcas. Maybe.
and also we can help. We can also help.
Yeah, we will help
Yeah.
her.
If
that's
you
the
can-
All right,
quickest
thank
thing.
you.
Yeah.
We will split the data into half and we will work it up each other. Prithvi, can you stop sharing?
Oh, yes.
Because we see 2-3 pictures, huh?
Yeah, 2-3 pictures.
Team has grown.
Yeah. Okay.
Dorcas, do you have any specific doubt with respect to data cleaning and other activities that we are planning to do?
No, not at all. If I have any issue during the course of the analysis, I'll raise it on the WhatsApp group.
Okay, okay, yes.
She can
Alright,
also share
thank
her
you.
files with us, because if we have to help, we need the files. Maybe like if she can edit the metadata file, I can try sit with the aggregate summary file. I think more work has to be done in aggregate summary. Individually, we have to edit the values. So because we have to simultaneously edit 2 files separately first, and then we have to combine it. So if she's comfortable with the metadata file, and I can work on summary file, or if she wants to edit summary file, I can sit with the metadata file.
Yeah, that's a good idea to split it. Yeah.
Yes. Please coordinate Prithvi with Dorcas, and we will help her.
Yeah. Okay. Sir.
Okay. And what I said, should we schedule another meeting in 2 weeks? Same
Zavo,
day, same.
Zavo has anything to clarify?
Yeah. Should we have a deadline for this one? Something like that. What do you think?
Yeah.
2 weeks time or 3 weeks time?
Yeah,
Jay, can we
makes
ask
sense.
with the other 2 ambassadors, Zavo and Mark? They are also here. They haven't spoke anything. Maybe shall we give an opportunity for them to?
Yeah, of course, everyone is welcome to talk. Huh?
Hello, Mark.
Hello, everyone. I hope you can hear me.
Yes,
Yes.
we can hear.
I don't have any problem. Maybe just ask when we finish, maybe the cleaning of the metadata files and the outputs. Where do we put them? Well, we shall still use the Google folders we shared last time. I don't know.
I didn't get Mark clearly. Mark, can you repeat?
Last time we shared the outputs we got to the Google
Correct.
Drive.
Yes, yes.
Yeah, so this time, maybe after cleaning the metadata, where shall we transfer the files that we've cleaned?
Yes, please put it into the same folder that Jay has shared after the cleaning.
Okay.
Yeah. You have the metadata file with you. I believe that you have it. You have already downloaded.
Yeah, I still have the raw files, but yeah, I'm now working on the new part that we're doing now.
Yes. Perfect. Perfect. Do you have any input suggestions to us?
At the moment, not so much, because I've not yet worked with quite complex data to this magnitude. So, yeah.
Okay. Yes. Yeah. Thank you, Mark. Thank you.
Shall we say in a deadline in 3 weeks, and that we schedule another meeting in 3 weeks?
Yeah, I think that's perfect.
Yes, I just saw the Google Drive.
Sorry, I have a question.
Yeah, please. Please.
Lucas, please go ahead. We can't hear you. We have to unmute your microphone.
No, you have to unmute your mic. Dorcas, your mic is muted.
So, okay, I have a question. Can I go ahead? Can you?
We can't hear you.
If you have a network issue, you can type it in the chat.
Hello, can you hear me now?
Yes, right now we can hear you.
Hello.
We can hear you. Go ahead.
Can you hear me?
Yes, we can hear you.
Marcus, do you hear us?
No.
Can you please
Dorcas,
type your question?
Hello.
Type your question, please.
Can you hear me?
Yes.
The answer is
Okay. I'll send a text message. You can hear me now.
Yes.
Oh, I guess it's the network. My apology for that. Yeah, I can hear you. Can you hear me now?
Yes, we can hear you, but
Yes.
Okay, let me try. Okay, my question is this. From the presentation that Krishna did at the beginning of the meeting, please, are you hearing me?
yeah.
Yeah.
Okay, there was an aspect where we were comparing clinical isolates with environmental isolates. So I was just thinking that why are we just using just two components of the One Health, just two components of One Health? Why not the three aspects? I mean, why didn't we include the animal segment of One Health? So I want to know if we have any particular reason for choosing just the clinical and environmental, for comparing just two segments instead of the three segments.
So if you see the updated SOP that we have shared, it has 4 segments right now. It's not only human and environmental, that is clinical and environmental. Right now, we also have it from animals. We also have it from plants. So there are 4 segments. So the
Okay.
SOP has been updated to have 4 segments of One Health component.
Okay. I actually saw the other components in the SOP, but when you were doing the presentation, you only presented 2 segments. So that's
Only
why I'm
2 segments.
asking.
Exactly.
Oh.
After the discussion with all the ambassadors as well as with these experts, we thought that we also need to include those other 2 segments to make our study more appropriate. So that is what we have added. So if you see our SOP that we have shared it, we have from the clinical, that is from the human, the animal, that includes all your livestock. Then we have it from plants. We also have it from environment. Environment includes water, sediment, soil, those materials. Yeah. So we have all.
Thank you.
Yeah.
I have to leave in a
Thanks.
few minutes because I'm having an external meeting.
Yeah.
I would like to round off, and otherwise I have to. I leave it up to you.
Yeah.
I
Just
think
one point can I make?
Yes.
So the Enterococcus faecium, that data has not been uploaded to the Google Drive. I don't know if that team has already finished that first part of their analysis or not. Because I tried to contact Bruno once, and I couldn't, contact him again. So I don't know. Only one that one species is remaining. Other than that, I think everyone has finished the first stage of analysis, and they are now moving to the second step of work. But I think the first one species, I think it is pending. So I think we have to check that also, because once we move forward again, coming back to from beginning, it will be difficult. So I think we have to track that one.
Yeah.
So that's the one point. Thank you.
Maybe Martin, you can write an email to those 2 colleagues if they want to respond, because as Priti mentioned, we cannot go back at one point. That'll be very difficult. So Krishna or Priti, can you share the names of the colleagues whom to Martin?
Yeah.
So let Martin write a detailed email or a polite email. Let us see how the response is.
Yeah.
Then we can decide.
It's Bruno and Shamsul Ansari.
Bruno is from Italy, who was
Please
also
share
part of the other
email ID
circle.
as well. Please share
Okay.
their email ID as well.
No, sir. I just have the list.
Samsul is working on the metadata. Martin, do you have any information from Samsul?
Yeah, he's originally from Nepal, but he is now in Japan. And I heard that he had some family issues, so that's why also is not that much involved. But we can contact him anyhow.
Okay, so Bruno and some.
Yeah.
Yeah.
Krishna, I will take this up separately with you, and most happy to send emails. And I will also share the recording, and I also will set the next date in 3 weeks, if you agree.
Yeah,
Yes.
perfect.
Please make it 4 weeks. Let us make it round.
Okay. Okay,
4 weeks,
perfect.
that would be really good. Okay.
So I will.
1 week cushion time.
Yeah. Correct.
Okay. And the final text I will also check with Krishna, and then we okay.
And how about the plans for the internal reviewer? How and when he will be coming inside?
Has not been discussed yet.
Okay.
He can. He's flexible, I'm sure.
Okay. Wonderful. Wonderful.
I think let us keep ready our clean data.
Yes.
Yeah.
Yeah.
I think within a month time we will have those in our hand. Yeah.
In our hand. Okay.
Yeah.
Yeah. I will come back to you, Krishna, with respect to the text of the email, that we make it very clear to all the ambassadors.
Sure. Okay.
Okay. Thank you very much. I have
Thank you.
I have to leave
Thank
now.
you.
Thank you. Thank you so
Have a
much for
great-
coordinating. Thank you.
Have a
Thank
great
you.
day. Yeah. All the best.
Thank
Bye bye.
you. Bye.
Bye bye.
Bye.
Okay.